Excellence is not an accident, it's a result of high intention, sincere efforts, intelligent direction, skillful execution, and the vision to see obstacle as an opportunity, as rightly said by Aristotle, an ancient Greek philosopher. So we are constantly looking for a speaker who are excellent and expert in their field of expertise. Our research came up with Dr. Mrs. Ruksana Pinjari, who is joining us today as a resource person for second session. In her talk, she will focus on plant derived natural product as a drug discovery. Prior to that, I would like to take this opportunity to welcome and introduce Dr. Ruksana Pinjari. Ma'am has received first class in graduation from University of Pune in 1987, a master's degree in pharmacognosy and phytochemistry from same university in 1992, and doctorate from the same university in 2016. Ma'am has huge experience of 29 years, of which she worked as a production trainee with New Life Pharmaceutical in Pimpri from 1987 to 1988, as an hospital pharmacist at Jizamata Hospital in Pimpri from 1988 to 1990. She began her academic career from YB Chavan College of Pharmacy in Aurangabad as pharmacognosy lecturer from 1992. From onwards, ma'am has hold various positions in pharmacy, including associate professor, department of head, CEO, professor and associate dean at Alana College of Pharmacy, at STE Societies, Srimati Kashibai Navle College of Pharmacy, Ponwada, Pune, school and at School of Pharmacy, GH Raisoi University. She is currently working as a professor and principal at STEC, Sinhgad Institute of Pharmaceutical Science, Lonavala. Her area of research expertise Her area of research expertise is natural product research, in which she worked on phytochemical and pharmacological behavior of medicinal plants in the area of anti-cancer and anti-diabetic research, development and evaluation of herbal formulation, isolation of phytopharmaceuticals. Her research is also funded by various agencies, like from Ayush Department of Ministry of Health and Family Welfare. She received 20 lakhs. From BCOD University of Pune, she received 2 lakhs. And from Maharashtra University of Health Science, she received 25 lakhs. The outcome of her extensive research is 37 research papers in international journal. Under her guidance, 31 MPharm students has completed MPharmC and 6 PhD research scholars has successfully completed their PhD. Ma'am has presented 12 research paper in national and international conference. Ma'am has authored 2 book and 3 book chapters. On her credit, 5 different patents are there and 3 copyrights. As far as achievements are concerned, she won state level research award in Avishkar 2015 in teachers category from Savitribai Phule Pune University. A best professor in pharmacognosy. The name of award is Devang Mehta National Education Award in 2017. She is also awarded uh, by Mount Albert Central University, USA, the honorable degree of Doctorate of Science, that is DSc in Pharmaceutical Sciences, in August 2023, a Best Academician Award uh, in year 2023 by Center of Education Growth and Research, New Delhi, and International Outstanding Innovative Educator Award in 2024. That is the latest award by Green Thinkers India. She won various membership like IPA, SFE, APTI, member of Board of Studies from University of Pune, member of Committee of Unfair Myths from University of Pune. With this very short introduction, I welcome you, ma'am. I request, ma'am, to share your expertise on plant-derived natural product for drug discovery and take the charge of this virtual platform. Please, ma'am.
Thank you very much. Am I audible?
Yes, ma'am, you are audible.
Okay. Okay. So, thank you, Dr. Mari, for that wonderful introduction, the elaborate one, though, for a nice
Okay, ma'am.
words that you have given. At
Thank
the
you.
outset, I would like to thank the principal of Shri Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nashik, Dr. Amritkar, the coordinators and the IQAC coordinator, Dr. Pinjari, for giving me this opportunity, identifying me as one of the speakers for this national level webinar themed as Exploring Pharmaceutical Innovations, the Future of Pharmacy. I must congratulate the principal and the IQAC coordinator for the wonderful title that has been used for this national webinar, because this is the need of the hour, and the innovations have been happening multifold now, but we need to know what is the outcome of the innovations that has been coming, and how we as teachers or our students at the PG levels or PhD level, or for that matter even UG level, will be able to contribute to this concept of innovation, which is the need of the hour. There has been innovation happening, but then is it result oriented? Is it giving output? Is what matters, and is a point to think, and a matter of discussion, debate, and really a thought process involved. So, thank you very much for this opportunity. I would begin my session now. Uh, Under the theme of exploring pharmaceutical innovations, I have given my expertise as the natural product drug discovery. And under that banner, I am going to talk today about the phytopharmaceuticals journey from traditional healing to innovation is my talk, and which will contain the initial history or initial performance of the herbs within the Indian knowledge system or Indian healthcare system, the world healthcare system, and then we are going to go to the innovations that are happening in the pharmaceuticals or rather phytopharmaceuticals or the natural products. So I am beginning now. Is my screen visible? I have changed the slide now.
Yes, ma'am. You are clearly visible and audible. You can continue.
Oh, yes, thank you, ma'am. So we know we have got a big heritage of the traditional healing, traditional systems of medicine, complementary medicine, alternative systems of medicine. We've got the heritage, we've got the ancestral treasure, I must say, for this. And under this traditional and complementary medicine umbrella, there are many practices that have been happening for all these years. Like it had been, some are continued even now, but they have been happening for last 2000 years across. So we'll just go through what were the practices involved, and how we have come all the way from these practices to the active pharmaceutical ingredients derived from the natural sources, and how they have been used successfully as a pharmaceutical. So the journey goes from the phytopharmaceutical to pharmaceutical now. The practices involved were acupuncture. You know all these, but still I will just brief in a sentence or two. So acupuncture, as we know, that you just have to have a micro syringe inserted, and then it reaches the nerve system where it stimulates the nerve system and heals. So this is through the natural healing kind of. Then the chiropractic. This chiropractic is the body's ability to heal itself, and the treatment involves manual therapy, especially the spinal manipulation. So, where you have the spondylitis, or you have the stiff joints, or you have the frozen shoulder. So, these chiropractitioners, which are called as chiropractics, have been using this to ease out the pain. Kind of physiotherapy, you can say. Now we have given it as a technical and a scientific term as physiotherapy. So it nears somewhere to this. Then we have the third practice that was carried out was osteopathy, which is a branch of medical profession. This is still practiced in United States for your information. And it is a pseudo-scientific system of alternative medicine that involves again the physical manipulation of the body's muscles, tissues and bones. तर ते हाड सरकणे वगैरे ज्याला आपण म्हणतो तिथे ते प्रॉपर त्याला सेट करून किंवा बट फॉर दॅट टू डू this looks and this sounds very crude, but then for that you need to have a proper degree to be taken. I mean, the doctorate in the osteopathy or even a doctorate in chiropractic is required. So this is still going on in the US, where US says we know that US is technologically 20 times advanced, but then they are still practicing our ancient sciences yet. Then we have naturopathy. You all know naturopathy is about the naturals, especially the food, and food is the healer, is what they call. Then we have the Ayurvedic and Unani systems of medicine, alternative systems, Ayurveda, Unani, Chinese, homeopathy, which more or less, except homeopathy, Chinese, Unani, Siddha, all these ASU systems have got similarly the same concept, the same diagnosis and the treatment pattern, and somehow same kind of or similar formulations, I must say. So we know this herbs and the traditional medicines and these practices for long, like maybe around 5,000 years back, we have been practicing this. Now, WHO has accepted along with these practices, the use of traditional medicine, that is the herbal medicines. And around 170 WHO member states have reported the use of traditional medicine by their population. So WHO has got a tremendous role in having the acceptance of the naturals. So what's the philosophy of the traditional medicine? The knowledge, skills and practices based on the theories, beliefs and the experiences indigenous to different cultures used in the maintenance of health and in the prevention, diagnosis, improvement or treatment of the physical and mental illness. So basically whatever has been mentioned in the Vedas, or then after the Vedas, then it came to the encyclopedias of the medicinal plants like Charak Samhita and the Sushruta Samhita. So Charak Samhita say around 100 BC or Sushruta Samhita say around 500 to 600 BC, where about Sushruta is the oldest one, where it has say around 186 chapters, 700 medicinal plants and 64 preparations. The mineral based and the herb based preparations have been mentioned. Since then you can imagine so it's around 2,500 years ago, where you have a scientific compilation of the herbs. So these herbs, people then have been using these herbs, referring to these books, like how we have pharmacopoeias now. So these books were considered as the official books by the what we can say is the hakims and the vaidyas, and refer it for the treatments, and they have been using it successfully, though the longevity probably was not as it is now, but then they have been using it to heal, to increase the life, to have the life comfortable and pleasant. They have been using it. So they have these compiled or compilations of the medicinal plants. These state the prevention, diagnosis, improvement or treatment of the physical and the mental illness is what is important now. So, what's the philosophy of the traditional medicine? It's the holistic approach to life, equilibrium of the mind and body, and the environment. So, we must also add yoga to this, and emphasis on health rather than on the disease, which is the concept now which has come back. Generally, the focus is on the overall condition of the individual rather than on the particular ailment of the disease. So, these practices, whether it is the Ayurveda or say Unani or even for that matter homeopathy, they have this individualism approach in it, which is now coming up for the modern medicine as well. So, it was one person, one body, one the setup, one balance, and then accordingly the medicine is being given. So this is the philosophy basically, but then it went on disappearing as the science progressed and we came to more technical know-how and more of the technical databases. So this approaches is lost somewhere, and now we are coming back to the same. So why herbal medicines? Now we are coming to the traditional medicines, or we are coming to the traditional or the naturals or natural drug discovery now. Why are they so popular? More affordable, as we all know, right? Tulsi is still used as antiviral, and it grows in your backyard or in your front yard. Chew few leaves of tulsi, and then you feel good. Or you smell the camphor, and your congestion is gone. Or you smell eucalyptus, your congestion is gone. And not expensive. You don't have to spend more than 100 rupees. So, of course, affordable. Corresponds to the patient's ideology. So you know that this is the plant which has been used by your forefathers, they who have used by where they had seen it, they had seen their forefathers using it, and so it's the ideology which has been developed that त्यांनी यूज केलं तर आम्ही पण करणार आहे, बाबांनी यूज केलं, आईने यूज केलं तर आम्ही करणार आहे, काइंड ऑफ. And same is passing on, which is a good thing. However, uh, yeah, again, it also highlights the concerns about the adverse effects of the chemical medicine. So it's sometimes feel that it's good to go with the herbals because they are safe, which is not true always, though we'll see it later. But then at least having one tablet, consuming in one tablet every day for say 15 days, one feels good to just chew a few leaves. Say for example, in my family I have two diabetic patients, and I have a gymnema in my front yard. So then, one of these diabetic patients of my family hesitate to take the metformin glipizide combination, saying that somewhere they've read and it has got the effect on the liver, and then it has the effect on the heart, and so and so. And what they do is they just chew few leaves of gymnema, and they feel that it is giving them the relief. So basically, it's the mental, what we can say, setup or ideology, as I said, which is working. But then again, it has its own pros and cons. I will come back to it later. Now that I've mentioned this gymnema, I'll come back to you later for that. So it satisfies the desire for more personalized healthcare. You go to the Ayurveda person or you go to the homeopath, and then they will give you those small packets or bottles or pudi, and then they will say you have to have it. So we have a satisfaction that this is made only for me, and it's not like a 500 milligram paracetamol or 650 milligrams of paracetamol, and which everybody is having without knowing the body set or the entire structure or even the ADME of that particular person. So this is the feeling of having the medicine personalized, as I said. And then there are some medicines which already have been told in Ayurveda as rasayanas for the health promotion. So the feeling is that, and the belief is that you have these medicines, or rather these rasayanas or these herbs, not medicines, rasayanas, and your health is going to be good forever. So there are these what I can say information databases of the traditional systems of medicine, which has made this particular herbal medicine, you say, or traditional medicine, you say, which has made it more popular. However, as I was saying, it is not necessarily true, especially when herbs are taken with prescription drugs, or they are taken along with the OTC drugs, or then the other herbs. Here I'm addressing the herb and herb drug and herb food interaction. So you are having it just maybe just few leaves of say tulsi or gymnema or licorice or cinnamon for that matter, but what you are eating along with also matters, and which can cause the trouble to your biological system. So, what are the challenges now, and why we need to go to the next level just from the herbal medicine? Why need to go in the little advanced stage, which is the need of the hour, of course. So, what is it? Lack of research data? Yes. Appropriate mechanisms for control of herbal medicines, which is not there, right? You can have maybe one stick of cinnamon, you can have maybe 4 leaves of gymnema, you can have 8 leaves of tulsi, but then how do you decide how many leaves is your dosage? How are they affecting your body? People have been having the madhunashini powder, right? And then they have it considering that it is used for the diabetes, but where is it going? Is it actually passing through your ADME process, or is it accumulating? Is it causing the load on the kidney? These are the points which need to be addressed before having them, or before the having the self medication of these herbal medicines. Education and training is required, of course, expertise within the national health authorities and control agencies. When herbal medicines are formulated, the regulatory authorities do come in picture. Information sharing. This is since long time. There have been the families and families who have been treating, these families of the Vaidyas or Hakims who have been treating the elements from generation to generations, but the message is not been passed on accurately, and hence there definitely are going to be after effects, side effects, adverse effects of these kinds of herbal medications which are taken by half information or using half information. Then it goes in hand with the safety monitoring and, of course, evaluation of the safety and efficacy. These are the main concerns when the herbal medicines are being used. WHO has a different say on this. They say that though it has been considered as pre-scientific and the treatments have been replaced by modern and the better efficient science-based medicine, what is less known is the contribution to the modern science and medicine and a long history of traditional products and practices being translated. When we will come to the innovation style, I will slides, I will tell you and I will give you the reference of this 6 number slide again, because this is being used in the innovation now. So we have been using the traditional medicine knowledge and then having isolating the phytopharmaceuticals and then the API formulating them. But this traditional knowledge is now being used for the different branches of the pharmacology and contributing to the innovations in the herbal medicine. Around 40% of the pharmaceutical products today draw from the nature and the traditional knowledge, including the landmark drugs. We are going to discuss these landmark drugs like artemisinin, aspirin, paclitaxel, etc. So these miracle drugs have come from the traditional system of medicine and the traditional knowledge itself. So we cannot underestimate the traditional medicine is the message actually. So just for your reference, everybody knows, but I definitely like to mention these whenever I talk about the natural product drug discovery. You see curcumin, obtained from turmeric, a wonderful antioxidant, and then all the related properties, anti-inflammatory, microbial, then the wound healing, and going towards the anti-cancer then. Licorice, again, hepatoprotective, immunomodulatory, the flavonoids being used as the reverse transcriptase inhibitors, etc. You have giloy, which has which was a hot topic during the COVID times, and because of its immunomodulatory property, Tinospora cordifolia. Then the poppy mentioned in Ayurveda. All of these are mentioned in Ayurveda. They are mentioned in the Charak Samhita. They are mentioned in Sushruta Samhita for the wonderful uses that has been giving. Now, poppy was used in Sushruta Samhita as mind soother, mind sedative, kind of. But then of course, whichever quantity it was given, it did not have the addiction, or it did not surface at least. But we know now that it has, as the technology enhances, so enhances the, malpractices. So poppy had been successfully documented as a sedative, as an anti-anxiety drug in Sushruta and in Charak Samhita as well. Vinca, a wonderful source for the anti-cancer drugs. You have Harda and the Amla for immunomodulatory property, and of course the cinnamon. Immunomodulatory property, all these three, the last three also anti-diabetics, antioxidants, and so on. So these are just few glimpses, I must say, which have been working wonders till US, who has not been accepting herbal medicine as a therapy or as a therapeutic agent, is being using now almost all of these ingredients. So now what's the journey forward? What is important? You cannot just have a maybe handful of powder or a handful of crushed crude drug and then make a kada out of it, is have, because there is no validation. One has a bigger fist, another has a smaller fist, and एका फिस्ट मध्ये एक मुठीभर घ्या आणि पाण्यात टाका आणि उकळा आणि प्या, is the thing which had been told by the practitioners then. But then एका मुठीत किती येत आहे, पाणी किती घेताय, उकळताय किती, त्याच्यातून ड्रग किती येत आहे, कॉन्सन्ट्रेट किती घेताय आणि किती पिताय, there is no validation, there is no measure, there is no mapping of how much is the drug going into the stomach, along with you are having with what are you eating with, what are you administering it with, right? So that is nowhere seen, and that is why then further compound isolation has come into picture. So the traditional medicine coming from the ethnopharmacology, or have the information of ethnopharmacology, and then the plants being subjected to the experimental screening, where you get the extract is then further put for the phytochemical and the spectroscopic screening to find the possible near possible component, and then that component being purified and then incorporated into the formulation and consider it as a drug, is what was the approach earlier. But now what is the approach? The modern approach is needed in that case, where now you have already the compound, right? And that compound needs to be subjected to further screening, biological screening, preclinical, and then it will go for the clinical, and then there is a development of a single component, and then you use it as an API. So this is the journey which is required. So halfway till the compound ethnopharmacology is supported, and then there is the computational chemistry which has taken this journey forward to have a pure isolated phytopharmaceutical. So here we come to the phytopharmaceuticals. Since we have been talking about the phytopharmaceuticals, so the phytopharmaceutical drug is defined as the purified and standardized fraction, as I told you earlier, with defined minimum 4 bioactive or biochemical compounds. See, when you talk about phytopharmaceutical drug, right, it can be a particular extract rich fraction, right? Say the alcohol rich fraction, or you can say flavonoid rich fraction, or you can have a compound itself and that rich fraction, the bioactive compound rich fraction, and it can have 3-4 compounds together, like how we have curcuminoids, right? So you have three compounds together, and that together are we can say subjected for the treatments and regarded as a phytocompound. However, when you talk about a single moiety, then it is an API. So phytopharmaceutical, when we say this term, it can have 3 to 4 components together obtained from the rich extract, right? Which has been used for internal or external use for human beings or animals, and it could be used for the diagnosis, treatment, mitigation, and prevention. Whatever is the definition of drug, what we are adding here is the component obtained from the medicinal plant, which is used for the diagnosis, treatment, mitigation, prevention of the disease or the disorder, right? Except the parenteral route administration. So this is the phytopharmaceutical, and we are going to go further with few examples. We know artemisinin, obtained from the Artemisia annua. We know catechin, which comes from tea, which also comes from the colas, which comes from, say, uh, St. John's wort. It is coming from catechu. We have caffeic acid, the phenol, water soluble polyphenolic acid is the right word, used as anti-cancer, camptothecin, camptotheca acuminata, curcumin, source is turmeric. All of these have been either used as antioxidants and subsequently used as cancer. And if you can see the status, these compounds are into the phases. Artemisinin is now artemisinin, and there are the derivatives artemisinin, which have been now uh, successfully used as a drug, basically. So, Cortem is the drug which is marketed now by Novartis and is being successfully used for the chloroquine-resistant malarias. Epigallocatechin gallate obtained from the green tea, again antioxidant, chemopreventive, immunomodulatory, etc., etc. So these are the phytopharmaceuticals. There are many examples. Again, epicatechin, genistein obtained from soy, lycopene from the tomato or from watermelon, paclitaxel coming from the Pacific yew, taxol bark, quercetin, which comes from many of the citrus fruits, and resveratrol coming from the grapes and the blueberries, raspberries, mulberries. We just feel that we've studied this maybe when you studied pharmacognosy or you taught pharmacognosy, but these are drugs of real importance which have marked a strong, I must say, a benchmark in the therapy, especially the cancer therapy, or say used as anti-inflammatory and anti-diabetic and so on. So they have been used and need to be taken forward for having the API incorporated into the pharmaceutical formulation and being used either alone or in combination. So some examples, again, as I told you, how the innovation has been happening. So from traditional medicine we came to phytopharmaceuticals. Now we are slowly going towards the innovation. So tapping the nature and indigenous knowledge to advance modern medicine. Here, for the compound artemisinin and its derivatives, we must give the credit to the Chinese scientist, to Yo-Yo, her team, who have isolated artemisinin. Before that, she has tried 240,000 compounds for to use as antimalarial, uh, which are effective against the Plasmodium falciparum, which is chloroquine resistant. I mean, it's not even responding for the chloroquine and the other quinoline derivatives which earlier were used in malaria. And then she was successful after using it from the sweetworm wood, which is Artemisia annua, and she could isolate this diterpene compound, and this became the. She also fetched the Nobel Prize for that, you know it, in 2015, because it had been, she has saved millions of lives who died, or these lives which died due to the resistance of to the antimalarial drugs, and she had done a remarkable contribution uh, to the therapeutics coming from medicinal plants. So she's already worked on around 2.5 lakh compounds coming from the synthetic sources, and finally she came to the traditional medicine database, where she found that there is one herb, Artemisia annua, which is being used in the fever, okay, chills and fever and inflammation. And she decided to use it for malaria, and she was successful at it. And rest is all the history. So these are the compounds which you can see artemisinin or diterpene. Then the companies took this forward, this compound forward, and then companies like Novartis and Bayer, they had their own analogs. And then these are the compounds which came in. There is another compound called artemether. Now this is the innovation, I must say, because it has given us the compounds, this artemisinin has then is been semi-synthesized, and we have got all these compounds further for effectively to be used in against the Plasmodium falciparum. And this all of these artemisinin, of course, is being used. Artemether, with the actual brand marketed product is Coartem of Novartis, and used in combination with lemifantrin for the treatment of acute uncomplicated malaria, and is very effective against the Plasmodium falciparum, acts against the erythrocytic stage of Plasmodium species, and also includes the infections acquired due to the chloroquine resistant areas. So, artemether, a wonderful drug again, a semi-synthetic derivative of artemisinin. Artemisinin is isolated and then semi-synthesized to get these compounds successfully cleared by the FDA, and then subjected to the market, and then, of course, gone for the after all the regulatory ups and downs, gone for the commercialization. Another is artemotil, again effective against Plasmodium falciparum, semi-synthetic derivative, acts against blood-shed lymphocytes, and very fast acting. So biological half-life is very good. Another is artesunate. Artesunate is another acetoxic compound of artemisinin, which WHO has recommended as a first line treatment, is more hydrophilic component of artemisinin and is being used successfully. So this is about artemisinin, how it has come from the herb and now it has been used as a proper API, put into the dosage form and then used against different species. Another willow bark, you people know about it, but just a little mention I must say. Willow bark contains salicin, we all know. And then the salicin, the bark extract was being used for, again, treating the inflammations and treating the pains and the inflammations and the fevers. And then the scientists started working, and then the Sumerians and Egyptians have been using it like long back, Mesopotamian era, 2500 BC to 3500 BC to 2500 BC. They have been using it for this, also using it for the to ease the childbirth, to ease the pains, etc. By the Greek practitioners. Then in 1897, Bayer, the scientist or the chemist Felix Hoffmann, then synthesized the aspirin from the salicin. So he first isolated salicin, and salicin is a glycoside, and then it goes in the stomach, and then it hydrolyzes, and you get a compound as salicylic acid. And then the salicylic acid also had the similar properties as acetyl salicylic acid, and this salicylic acid was then acetylated, semi-synthesized, and aspirin. And we all know how miracle drug is aspirin, starting from the anti-inflammatory to analgesics and anti-inflammatory non-steroidal drug, and then going to say for the improving blood pressure, and using in the cardiovascular systems, atherosclerosis, and now there is no looking back, blood thinner, etc., etc. And it has come from the ancient 3500 year old bark. So we need to respect the heritage that we have and then work forward, is the message actually. These are following, see the brand names, and here is the market. I don't want to go into that. I just thought that, look at the percentage captured by aspirin in the market. 2023, 70% market has been captured for pain and fever. Rest 10 and 20%, say 20% for cardiovascular and others. In the by 34, this market is going to increase by 10% at least. So this is going to go on. Here is another miracle drug, and that is Taxol, which comes from the Taxus brevifolia, and various species and various derivatives of Taxol has been used. What I want to tell you here is, I will not take more time here, because I have to also talk about the other aspects. So, see, paclitaxel, when initially was synthesized, and was isolated and not synthesized, sorry, isolated in 1971 by the Pacific yew, or you can say the Taxus bravifolia and the Baccata and the rest of the species. It, when was marketed, the price was not less than $200 initially, right? US dollars. Somewhere in United Kingdom, it was same 1,500 to 2,000 pounds was the one vial of maybe 10 ml costing. Paclitaxel as a compound. It has got a very good anti-cancer property through the antimitotic mechanism, and is been recommended by all the physicians as for the ovarian cancers, for the prostate cancers, etc. Very effective with the minimal, or we can say bearable toxic effects. But then so much of work of the scientist has happened now with the, you can see all these derivatives which have come one after the other, and now the cost of the product is not more than say 7,000 rupees. Say paclitaxel 100 milligram 100 milligram vial. This is given parenterally, of course. So 100 milligram containing vial is costing 5,881. So Surya Specialities is the brand. Then another brand is again 100 milligrams. It's just costing 3,200 now. There are compounds. There are certain companies which are also selling it for 100,000 rupees, as against the prices of the other anti-cancer drugs, which cost not less than 21,000. This is the minimum that I'm talking about. 21,000 for one dose. And then you know how many cycles you have to take for the for treating the cancer as a therapy. And paclitaxel, in comparison, is, I must say, 10 times lesser and cost effective and affordable to the common man. This has come through the innovation. This has come through the research and innovation. Right? So, the National Institute of Health and Care Excellence has recommended paclitaxel now. It has been now accepted worldwide, and it has been used for the breast cancers, and for the ovarian cancers, pancreatic cancers, Kaposi's sarcoma, where you have the lesions, or you have the tumors, or the proliferations into the inner lining of the blood vessels, or even on the skin cells, etc., etc., and in the alveoli, lung cancers, and is effectively being used with the lesser side effects. So this is something which is remarkable about the paclitaxel, and because this has achieved that benchmark of having the cost price reduced, and so reduced is the toxicity, I think it is important to mention this compound here. Similar are the products, say Ralphia serpentina. You can see there is a structure activity relationship, and these drugs have been used now effectively. Risapine has been used. Desapidine has gone into the phase 3 clinical trials, though no compound yet has come up. Vincristine, we all know. There are the semi-synthetic derivatives of vincristine and vinblastine coming from Vinca, Catharanthus roseus, and having the semi-synthetic derivatives of vendacine and venorelbin. The compounds are marketed, and the very popular brand that is being used is the Javlor, 25 milligrams per ml concentrated injection of vinflunine ditartrate, which is being used for the child, even in the childhood leukemia. So again, a benchmark attained by a normal ornamental plant, and a good example of how the innovation in the herbals has happened. Strychnos vomica, considered as a poison, but is the acetylcholine or cholinesterase inhibitor. It is also used as the antispasmodial, can be effectively used in malaria in that case. Also has got some anti-cancer evidences. So the compound is poisonous, but yes, the work can definitely go ahead and is been happening at the National Institute of Health, USA, for the analog being less toxic and more effective. So maybe in 10 years down the line, this compound also may come in its derivative form to be used maybe as anti-cancer or as another therapy for the anti-malarial or so on through the phytopharmaceutical innovation, I must say. So, what's the innovative approach? I just spoke about it that you have the herb, right? Go in for the extractions. Extraction, then you have the rich extract, go through the spectroscopic studies and find out the new molecule, and then have the applications of new drug done. Go in for the preclinicals first, and then the clinical trials, and then having it as the pure API and use it. So this can be done through the experiential phase, exploratory phase, and these further studies for getting the lead molecule. So I will not go into the depth of this because this is pure pharmacognosy. So we will go ahead. This slide I like because this talks about the various innovative branches that come or that support the medicinal plants for their further innovations. So you have if you see here, you have a polypharmacology, you have network pharmacology, you have reverse pharmacology, and then you have which is coming from say ethnopharmacology, high throughput screening. So these are the few important terminologies which support the innovation in the medicinal plants, or you can say the herbal medicines for that matter. So this slide explains the identification or say discovery of a compound or a group of compounds or a phytopharmaceutical from the medicinal plants through, as I told, exploratory and the experiential studies and the learnings. So let us go to supporting to this slide. Let us go to the few branches. You have already know you've been studying these branches. I'm sure all the pharmacology people know about it. Even the other branches know about this. But I want to talk about these particular branches with respect to the medicinal plants. So this reverse pharmacology is coming backwards, right? Going from the laboratory to clinic, it is coming from the clinic to laboratory. So the drug is being known. Like, see, we can here give the examples of artemisinin or even for of the willow bark for that matter, which had been used already for a particular indication, and then the compound has been isolated and then incorporated as a formulation. So these are very good examples of reverse pharmacology where you are not taking that conventional path, right? Having the traditional knowledge, then going extraction, and then going further isolation, and then somewhere finding out the component, and then going in for the clinical trials or finding out the biological trials, finding out the bioactive component, and then elucidating the structure and going further, right? So for the NDA and ANDA application, and then the lead and the drug. So what this is doing, this knows that there is certain compound which is being used for a particular related ailment. So then that is targeted to screen biologically for that particular area only. It doesn't have to hit the random areas of the therapeutics, right? It can go only to that particular area. For example, as the Chinese scientists have done, Yotso. So what has she done? She saw it that it is being used for the chills, for the high fevers, weakness, fatigue, and she decided to use it against the plasmodium species, and she was successful at it, and then she took the matter ahead, right? Same is with the willow bark, where you have salicin into it, and then the convergence, and then semi-synthetic derivative as acetyl salicylic acid, and then the further derivative. So these are the examples of the reverse pharmacology, where you know what the product is being used through the traditional knowledge that you have. So hit only that particular biological area and try and find out the lead out of it. So this diagram will give you the clear picture. Conventional method, you isolate, then target identification, lead optimization, preclinical studies, clinical trials. Now this compound is already been isolated. Just go in for those biological screening and go directly for the clinical trials and then give it for the approvals, because it already is been approved for a particular indication. It has been already proven for the toxicologies and the pharmacokinetic and pharmacodynamic parameters. It saves the time. So if the drug is coming to the market in 8 to 10 years in the conventional way of drug discovery, 8 to 10 years is minimum, it goes to 8 to 15 years. It can be reduced to 4 to 5 years just through the reverse pharmacology. And this has been the popular most branch that is being used now to save the number of years of the investment in terms of personnel, in terms of money, in terms of years, in terms of all the consumables and so on. So here is, sorry, just one example, one or two examples we can take. The Parkinson's had been used. Muconopruriens is a plant which also contains L-DOPA, right? And it has been already known that it contains L-DOPA. This maybe the needles present in these muconopruriens is it causes the itching. Like khaj khujli is what is the plant known for. But it was found that it contains L-DOPA, and we all know that L-DOPA directly, or the strength of L-DOPA directly affects the psychomotor, or even the the mobility of the person, the neuronal system of the person, right? And then they used it for Parkinson's disease and were successful. So they worked with the Jandu laboratories and they produced a drug called Zandopa, which is successfully being used in the Parkinson's disease now. So there is some cure, you can say. So muconopruriens, it was proven that it contains L-DOPA, and Parkinson's L-DOPA has everything to do with the Parkinson's disease. I will not go into the mechanism. And then so is the correlation which you get through the reverse pharmacology, and then come back with the new drug effective against a particular disease which requires a lot of research. Another, again, if you see, psoriasis. The psoriasis was this is a disease we all know. It is considered as the autoimmune, and then it cannot be treated, and is what being told. So this is being used for, this was done through the the Lupin laboratories use this for the plant for the treatment, and then the drug candidate Desoris was used, and this was obtained, again the formulation obtained through the reverse pharmacology. I will tell you the drug. I'm not immediately getting it through my head, but I'll tell you what the drug was. And then this has been used. There was a proper new drug application put in, and then the drug came up through the 5 to 6 years, and a very good drug called as Desoris is now being used. It's the oral formulation and not the application. The gels are also there now, but it is through the oral formulations which is being used, and it actually softens the tissue. The scaling goes off, and then the dead skin falls off, and then there is a new regeneration of cells and so on. So this is a wonderful drug that has come up for psoriasis through the reverse pharmacology. Another branch that is being popularly now used by the scientist is a polypharmacology branch. So polypharmacology, where a molecule is designed to bind to two or more targets simultaneously, such that the combined effect has a greater therapeutic outcome than binding to just one target, is what is the polypharmacology. So we know that one drug, one target. The drugs have been used, even if the compound isolated from the medicinal plant or from the allopathic drug or API, synthesized or synthetic API, we have a concept of having one drug, one target. But polypharmacology talks about the one drug and multiple targets. So when it works on the multiple targets, the efficacy is improved, the dosage can be reduced, and the drug can be most effectively used for a particular ailment. The polypharmacological profile of five natural compounds, curcumin again, which we've talked about before, epigallocatechin, which comes from tea. These are the polyphenol, water soluble, high molecular weight polyphenol, epigallocatechin, gallate. Quercetin, again, a flavonoid. Resveratrol from the grapes, blueberries, blackberries, raspberries, strawberries, and of course the grapes. A compound with the wonderful antioxidant activity and then now the proven anti-cancer activity for face, the berberine from Berberis aristata. These compounds now are being used for the multi-target therapy using the polypharmacology effect. So if you see this diagram, you have a compound and then it shows the efficacy against target 1, target 2, target 3. For example, if you are taking resveratrol as the anti-cancer drug, then it should work not only say on the sarcomas, or it should work on the lymphoids, it should work on say, and these present into different organs. So at a time it should work on three different targets or three different organs with three different binding to the proteins. So the effect is multifold, and again the cost should come down because the efficacy is more, the dosage reduces down, and the cost automatically comes down. So this is just a superficial explanation of how polypharmacology, where one drug can affect the three targets and achieve the desired effect, can be used. Another branch of the I should say innovations in the traditional medicine or herbal medicine or a point of natural drug discovery is the network pharmacology. So the network pharmacology is something where a multi-target drugs can be used against complex diseases such as say diabetes and cancer, uh, and is now fast growing in the approach in the drug discovery. Regarding this, the network pharmacology defines disease mechanism as network base targeted by multiple synergistic drugs. The use of network pharmacology to better understand the mechanism of action of herbal medicines. This was first uh, proven in 2007 by Hopkins, where he termed this coined this term as the network pharmacology, which is based on the idea that several highly efficient drugs act on numerous targets rather than just a single one. So you have the two, three compounds together can be, so more number of compounds and more number of targets basically can be used. So see, you have, there are, this may basically works on the symptom, you see. Say for example, so there are three symptoms and then there are three compounds together which will be working on three different proteins and then can achieve the result. Say for antihypertensive drugs, just an example, antihypertensive drug, say it could be the ACE inhibitor that you have been using. So what it does, it just dilates the vessels or then blocks the receptors, as I told, ACE inhibitors, or it just causes the dilatation of the blood vessels and then release the pressure and the hypotension occurs, right? But is it going to the root cause? It is not, or it is just working maybe onto the blood vessels, right? But then what about the brain, which is causing the stress and which is causing the narrowing of the blood vessels, or the lack of oxygen is giving the pressure, right? So then there could be a drug, a combination of the drugs, or a single drug even, can work on three different sets of proteins, three different receptors, which will be working on three different targets and achieve the result, right? So then it is going to, I must say, work towards the root cause, right, and not the symptoms. So we know when you have a blood pressure, you feel uneasy or you have a breathlessness and so on. And then we don't know the actual reason. We just know the symptoms, whatever has been happening due to BP. But then are we addressing the root cause or we are addressing the symptom? Symptom, if it is addressed, then you have the dilation of the blood vessels and then the pressure is released. You feel temporarily okay, right? But then what about the reasons, other reasons? Is the brain affected? Is the lung, are the lungs affected, right? Is your gastrointestinal system contributing to the increase in blood pressure? So you have three different protein targets and then you uh, bind there to those three different proteins and then achieve the result of dilatation of the blood vessels, reducing down the stress, improving the supply, oxygen supply to the lungs, and threefold contribution to the reduction of the hypertension. So this I can, I guess, explain well with this hypertension example through the network pharmacology concept. We go to the last concept, or other second last. There's one more thing. I think I am exceeding the time, but just 5 minutes more. I am still audible, right?
Yes, ma'am, you are audible.
Okay, okay, all right. So next is the nanotechnology. Now this is a huge topic where you can talk for 2 hours, but I just want to give you a few examples of the innovation that has happened in the new drug discovery of the medicinal plants or the phytopharmaceuticals, which is addressing the bioavailability, which is also addressing the selective binding at the receptor sites. It is also specifying the targeted deliveries of the phytopharmaceuticals, and then achieving the result that we want. So solubility, we all know curcumin, for that matter gymnemic acid, for that matter artemisinin, or even though we say that water soluble polyphenol, still the biological half life is less, the absorption and the biological half life is less, and then the impact that it should give for being used as, say, free radical scavenger, or then subsequently as anti-cancer, or say anti microbial, or whatever is the antiviral, anti microbial, whatever is the effect that it is desiring, is not going to happen. So if the solubility is a concern, we all know curcumin. We have been reading a lot about curcumin's poor availability and how it has been worked wonders through various approaches of nanotechnology now, reaching out to the targeted site for the cancer treatment and also for the inflammation. So, nanotechnologies, as I told you, is giving you the improved bioavailability, selective receptor binding, targeted drug delivery of the phytopharmaceuticals, and is one of the best innovations that has happened even to the herbal medicines for that matter. This we have already spoken about, so I will just skip the slide and go ahead. Yes, as I told you, that the compounds, phytoconstituents, or the phytopharmaceuticals, due to the poor water solubility and the low bioavailability, needs to be incorporated into a particular delivery tool, which is going to take the drug to the absorption site and then open and release the drug, so without affecting all the biological environment. These are the different nanotechnologies that are available. We all know, you know about it, say liposomes, dendrimers, solid lipid nanoparticles, metallic nanoparticles, nanocapsules, nanocrystals, nanogels, nanoemulsions, etc., which are being used. We are just going to talk about few examples. These are various nano drug delivery systems. You all know liposome, neosomes. I will not explain them because now that is not my area right now. I just need to introduce these to you to address the innovation in the medicinal plant. So liposomes, neosomes, phytosomes, self-assembled micelles, right? Polymeric nanoparticles, gold and silver nanoparticles, silica nanoparticles, SLNs, nanoemulsions, nanostructured lipid carriers. These are the many nano formulation platforms or nano formulation or nanotechnology called platforms that can be used for synthesis of pharmaceuticals,
Hello, ma'am. Ma'am, are you disconnected? Hello?
I told you about the
Yes.
uh, curcumin loaded, see liposomes. We have curcumin loaded cyclodestrine, gamma cyclodestrine liposomal nanoparticles being used for in breast cancer, in lung cancers, and the outcome is apoptotic cell death in the breast cancer cell line and at A549 cells. Then you have quercetin. You also have the gallocatechin gallet in the lung cancer, quercetin in breast cancer, voganin, say you have it here. It is in combination with the polyphenols, which has been used in the liver cancer and effectively used in as an anti-cancer drug, uh, affecting or targeting the hepatic cancer cells. So this is about how liposomes are being used, how they go. I had the videos which I wanted to actually show, but I think it's going to take time. So again, there would be a problem of sharing, so I will not have the videos shared. So just the tabular examples of nanoemulsions where catechin is being used, paclitaxel is being used for the prostate cancer or brain cancer. Outcome is apoptosis based inhibition of the cancer cells, prostate cells, cancer cells. You have the brain tumor uh, effective, which is 2.5 times more effective than the paclitaxel you see here. So imagine if paclitaxel has a less solubility and you put it into the nanoemulsion form and the efficacy is increased 2.5 times more, the cost comes down and so comes the down the toxicity. So this is something like really important. Then you have curcumin particles, self-assembled nanoparticles, which is being again used in prostate cancer. So if you see curcumin being used in breast cancers, used in the brain tumors, used in prostate cancers, liver cancers, it has got a wide range of anti-cancer activities. And in the form, if you address the bioavailability and the solubility concern, it can work as a miracle drug. And as I told you, multi-targeted approach can definitely be utilized. Neosomes, right? You have lauzone obtained from henna. We never thought that lauzone, which is obtained from henna, also could be effective in cancer. But yes, it has gone to the phase 3 trials, effective in breast cancer and MCF7 cancer cell lines. Again, lauzone, when alone is being used and when it is used as neosomes, has got 1.3 times more efficacy. Paclitaxel again, which is obtained solubilized by parenteral nutrition nanoemulsion when incorporated with the parenteral nutrition nanoemulsion used in brain cancer. And again, the efficacy is 2.5 times more. Same is with the phytosomes of resveratrol, where it is used in prostate cancer, and the activity is again twofold more. This is about the ones that are used. You have a session at 1:30, so in 2 minutes I should wind up. Drug repurposing, something is a very important approach now to the drug discovery of the traditional medicines, where you already see in the toxicity and all the ADME parameters of a particular drug. So you are not going to waste 4 to 5 years in just studying the ADME pattern, right? So you can skip that and just have it targeted to the different indication, right? One very good example of drug repurposing, which is not from the medicinal plants or the traditional medicine or naturals, is the use of metformin, which is being used as anti-cancer after repurposing. So metformin is being clinically tested for all the parameters of ADME. All the toxicity studies are already done. We know that compound is being uh, safely used with minimal to minimal side effects, also those which come in the prolonged run, not immediately. So this is already done. But accidentally, we get to know that the people who are using it for the blood, for the hyper or hypoglycemia, or used for the diabetes, type 2 diabetes especially, when it's being used, had when they were tested, it was seen that the subjects also showed the what we can say is the anti-cancer property. And the cancerous patient who also had the sugar and they were treated with this, it was seen that the proliferation comes down, the apoptosis goes down, the cytotoxicity is lesser, right? And then, or rather, the drug, or we can say the proliferation is coming down. So basically, apoptosis. And then this came as a miracle. And then now metformin is being addressed for the cancer as well, especially for the prostate cancer and the lymphosarcoma. It is being, I am yet not sure whether it is marketed for cancer, but is under the phase 3 clinical trial. So you can see how much time is gone. All 4, 5, 6 years are already skipped off because its ADME is already known, and it is being used safely as anti-diabetic. So it can definitely be used as anti-cancer. Only maybe 4 or 5 years further dedicated can make this drug as a wonderful anti-cancer agent. So there are certain databases, like how I gave you the example of metformin. There are certain databases. See, these are the databases. I will not read them because it's taking time. So various databases where you have more than 200,000 compounds. So you know that they are safe. They have been tested pharmacokinetically, pharmacodynamically for all the toxicity studies, all the ADME studies, and they are safe. So take that drug, study it well, see what is the history of it, what it has been previously used for, and can try using it for a particular indication through various preclinical and clinical trials, and go ahead to make a new drug with the new indication. So this is something, a very new branch, and which I am more attracted to, and I am working on this now, drug repurposing, and soon I will come up with some publication through this. So this is what is being told. See, just last example and we spot examples of approved drug and interaction of drugs to these target proteins. Okay, so you have the example of galantamine, which is the ACE inhibitor, but is now being used in the Alzheimer's disease. We already know that this is a very good ACE inhibitor and can be used as a very good antihypertensive drug. if it is used in dementia, and the trial is going to take only 3 to 4 years, and it has taken only 3 to 4 years, and drug now has been recognized and is approved now for the Alzheimer's disease. Right? Same the chinodeoxylic acid, which is used for the hypercholesteremia, or for reducing the cholesterol levels, and thus atherosclerosis, and then the hypertension and the cardiovascular diseases, etc., etc. So these are various. See, you can see various targets that can be used, and which already is been. I did not put the data here for what were their ADMA parameters and how they were safe and what time it took. But maybe next time, if I talk about only drug repurposing, we can go elaborately through this. So, with this, I want to come to the end of my presentation. Thank you very much. I hope I have given the justice to the topic that you people have chosen. And thank you to the organizers for giving this opportunity to me to express my reading and my interpretation of what I have read to all you. Thank you so much. Hello.
Hello. Yes, ma'am. Actually, we are having technical glitch at our. Sorry,
No, no
sorry for.
problem. But I was audible throughout the session.
Yes, yes, you are audible, but within last 5 minutes, actually, we lost the connection.
Acha, acha, okay. So
Okay,
was
ma'am.
it from my side or it was?
No, no, it's from our
Okay,
side,
okay,
ma'am.
all right.
Okay, ma'am, we
Okay,
have got
okay, all
some
right.
few
So I'm done.
questions.
Yeah,
Yes,
please,
ma'am.
please. Yeah.
Yeah, ma'am, are you able to see the questions? No, you can. Just a minute. Yes.
You will have to read them. I can't see. I have not accessed the YouTube through my mobile.
Ma'am, can you see now?
Can you please read?
Yes.
No. I haven't, as I told you, should I minimize my slide and maybe I can?
Yes, yes, ma'am. Yes.
Minimize.
Yes, ma'am. We have got a question from Divya Ingre. She is asking,
Yeah,
in
please.
future direction, what are some current efforts or initiatives that aim to further explore the relationship between traditional medicine and modern pharmacology?
Okay, good. I like the questions coming my way. That indicates that people have been actually listening to me and not getting bored through the entire 1+ hour session. So, a nice question. How and what efforts you should take to go through, I mean, to go from the traditional medicine to the modern pharmacology. So, as I told you, these are the various branches that you can approach to. The best way, you know what, is to go through these databases. The databases are available, and if you're doing it on your college level, then you can have go through the traditional medicine database, try and find out what are the herbs which are easily available, and study their potential, and target the particular disease that is being mentioned in the traditional system of medicine. And the process will go through, obviously, like having it at the laboratory level, extraction, and then the fractionation, and then the isolation, and then purification, structural elucidation, and then the process will go on as the preclinical screening. And then if the results are good, then you go in for the clinical screening. So once the clinical screening is done, then you have a real challenge where you have to prove whether your drug is safe and not toxic or less toxic in the given dosage form, and then have the new drug application done and get your lead compound for the phase 4 trial. So actually it's a big process, you see. But if it is at the student level, even if you go to the active fraction, the particular solvent or particular component rich fraction, and go through the same data, you see, still you get the results, and your results are going to be useful for the other scientists, other researchers, your data, they can refer to your data and the work further can go on. This is one approach through the phytopharmaceutical. Another approach that I told you is through this is something called as reverse pharmacology only. Then you can go in for the polypharmacology for the multi-target therapy, or then you can go for repurposing, as I told you, drug repurposing. For example, paclitaxel is being used as anti-cancer, say, right? So you know that paclitaxel, though it is in the parenteral form, is used as anti-cancer. Are there any other areas which should be touched apart from the cancer? Because paclitaxel is known for cancer only. Can there be any other area where paclitaxel as a diterpene can be used, right? There are many other areas. It is HIV, autoimmune diseases, arthritis, osteoarthritis, where you have no cure. So can this be used elsewhere? So go into the database, see uh, whether it is being used, or even the plant is being used for any other indication, and go ahead with it, right? So either you use this, the drug repurposing, or you use your network pharmacology, you see, and can go ahead. And another, I should say, area that you can access is have your drug. For example, grab say easily available, separate out the resveratrol, make the semi-synthetic derivatives in the lab itself, right? And then treat, or and they go in for the biological screenings and see how effectively they are being used, right? So this is another approach that you can use for using traditional medicine or medicinal plants as the APIs or as the modern medicine. I hope I have satisfied the query.
Yes, ma'am. Now we have got another question from Professor Shelke, ma'am. She is
Ah.
asking how to choose biomarker for a very new herbal product.
See, ma'am, nice question. Now, when you talk about the biomarker, when you have a drug, herbal drug, you need to first go through the phytochemical studies, right? Preliminary phytochemical screening, I must say. When you go through the preliminary phytochemical screening and purify that compound, go for the fractionation, purify the compound, and then go for, say, the chromatographic techniques. Okay, you have various solvent systems specifically used for that particular class of the phytoconstituents or phytopharmaceuticals, right? So, if say for flavonoids or for tannins or for alkaloids, steroids, there are specific solvent systems which will respond to only for that particular class. Once your drug through the chromatographic techniques, TLC initially and then HPTLC, you get to know that yes, this could be the class of the compound which it is responding to. It is giving you the band for that particular solvent system. So it is the compound. Say for example, chloroform methanol glacial acetic acid, okay, or toluene ethyl acetate water, or toluene ethyl acetate acetic acid used for the flavonoids, say for example, and a particular class, say flavonones, right, or the flavonols, it is being used. So you when your drug, your purified fraction is responding to this through this solvent system, there's a huge database for the solvent system. So you know that this could particularly be a flavonol category or flavonone category, right? Then choose the marker accordingly. So if it is a flavonol, you use quercetin. If it is a flavonone, you use, say, hesperidin, or also you can use maybe your catechin for that matter, and or gallic acid, and then check your or compare your findings. So the solvent systems or the chromatographic technique is the best tool which specifically can separate out the component on your stationary phase, and then accordingly you can choose the marker. That's the best thing. Suppose you have, you know, your compound is giving your the polyphenolic acid is being seen or is being eluted with the solvent system, right? So you can use caffeic acid or say gallic acid as a marker. So you need to first separate the component, or at least take the fraction, have the chromatographic data gathered, and then once your chromatography tells you that this could be the possible compound, then use a marker. Initially, if you do not know what the compound is, and if the compounds match, if your RF value is same on HPTLC, or the bands are similar and the color elutes are similar, you it goes somewhere nearby. You know the class of the compound, and then go in for the structural elucidation further, and it definitely gives you a compound that of your choice. Once you know the compound, biological evaluation becomes easy, knowing the behavior of the compound. Once you know the structure of the compound, you know how it will behave. Is that fine?
Yes, ma'am.
Yeah.
Ma'am, the last question.
Okay.
Okay. Last question is asked by Ankit. How do you manage immunogenicity risk with nanoparticle of plant-derived drug delivery? And what are the key challenges in emerging nanotechnology with plant-based natural product for drug discovery?
Immunogenicity. The nanotechnology still has not addressed this parameter. It goes to that particular target, right? And then it delivers its compound there, and then it behaves same as the normal API, how it should behave in the system, right? But immunogenicity is a very vast area, and it is individualistic, right? It will differ from the person to person depending upon its immunity status, and then this is addressed only through the individualistic therapy. So this will have to, this is again, I should say, going to go ahead. The further, what I must say, the innovation or further area of innovation on which it can be worked upon. The work is happening though, right? It is definitely addressing this particular area, but still I have not yet touched this area, but I will definitely go ahead and check about this. So nano drug delivery system, yes, it definitely works through addressing various parameters that we spoke about. Ghost makes the drugs bioavailable, and then it depends on the individual how much, and the individual system how much the drug is being absorbed and how the body responds to it, right? But then it differs from individual to individual depending upon their biological system, as you said, right? So this needs another, I should say, uh, approach to study. But this not necessarily is only for the nanotechnology. This could be for any administered drug, and can have the problem of immunogenicity. See, it could be our normal API, normal paracetamol formulation also. So nanotechnology definitely can address this problem, and can should address this problem in future. Definitely, this is a separate branch altogether, and a lot of work has been happening. Thousands of researchers are working on this particular area.
Uh, yes, ma'am. Thank you so much for satisfying the queries of our participants.
I hope the queries are satisfied, or else then they can mail me and I can explain them further.
Yes, ma'am, definitely.
Yeah, yeah.
Ma'am, as rightly said by you, we have actually a rich heritage of Ayurveda knowledge. So modern
Yes.
science and ancient knowledge should go hand in hand for better development of health of an people. Ma'am, you have highlighted on new techniques for development of active pharmaceuticals from herbal source like reverse pharmacology, polypharmacology, nanotechnology has discuss on case studies showing development of an APIs from such technique. Thank you so much. Uh, Ma'am, please accept this certificate as a token of appreciation from Gokhale Pharmacy College. Please, ma'am.
Thank you. Yeah, sure. Thank you very much.
Ma'am,
And Yeah, thank you to all the organizers for giving this opportunity, Mari ma'am, Smita madam, and all, Dr. Amrutkar sir, and all those are involved into this, into organizing this national level one day webinar. So thank you all of you.
Thank you so much, ma'am, for sharing your knowledge and valuable time with us. So, from Gokhale Pharmacy family, actually, I'm thankful to you. Thank you so much, ma'am.
Thank you. So should I leave now? I will
Yes, ma'am.
join another session, but now I should leave from here, right? Yeah.
Yes, ma'am.
Okay. Thank you. Thank you.
Thank you.